Alright, so here's my lesson 5 group problems. So for the first one, it gives us 3 genes. Mahogany, agouti, and ragged are the recessive, and then each one has a wild type, and we have to figure out the allele arrangement. So I looked at the two most numerous types, and I got agouti, wild type, and mahogany is one set, and then wild type, ragged, and wild type. So it looks like what is it? Part A in 1. And then the next thing I had to do was figure out the allele arrangement. So I looked at the two least numerous, the agouti ragged type wild type and the wild type mahogany. And I saw that the middle gene, the one that like switches, was the agouti gene. So that one goes in the middle. And then for part C, I had to figure out the distance between the agouti and the ragged gene, and then the agouti and the mahogany gene. And I did that by looking at as if the mahogany gene switched, and I did those numbers, which ended up, or the mahogany is 1 plus 1 plus 13 plus 12 over 206, which is the total number of progeny. Then I multiplied it by 100, and I got 13.1 map units. And then for the ragged, it was 1 plus 1 plus 18 plus 16 over 206 times 100, which is 17.4 map units. And then for part D, to calculate interference, you multiply your two probabilities, so 0.174 times 0.131, and you get 0.0228, and then you multiply that by the total number of progeny. So you expect to see 4.7 progeny that have double crossovers. And then the interference is 1 minus how many you saw, which was 2, over what you expected, 4.7, and you get 0.574. Multiply that by 100, and you get your percentage, so 57.4% interference. And for the next 3 questions, it was exactly the same process. For question 2, it was about the fruit fly Drosophila, and it involved the scute bristles, the sable bodies, and the vermilion eyes, and all 3 of those are recessive, and each one have their own wild type. Once again, I looked for the two most numerous, and I got wild type for the bristles, normal bodies, and normal eyes. And then for the other side of the heterozygous chromosome was the normal bristles, not normal bristles, scute bristles with wild type bodies and wild type eyes. And then to figure out the middle gene, I looked at this to see which one flipped. And I got that the scute bristles was the middle gene. And since it didn't say to figure it out for the heterozygous parent as it did in the other problems, I put both parents, both the heterozygous and the recessive, because it didn't ask just for heterozygous. For part C, I had to figure out the distance. It was the same process as before, so this ended up being 10 plus 14 plus 15 plus 156 plus 150 over 1,000 total progeny times 100. You get 33 map units between the S and the SC gene, and then from the SC to the V, it's 10 plus 14 plus 30 plus 46 over 1,000 times 100, which is 10 map units. And then I just drew out what the gene would look like. For part D, you multiply those again, so 0.33 times 0.1 is 0.033, and we had 1,000 progeny, so that becomes 33 we expect to see. And our interference is 1 minus what we saw, which was 10 plus 14, 24 over 33, which is what we expect. To get 0.273 times 100 is 27.3% interference. Number 3, just like the other two. That one is asking about the fruit flies again, and the phenotypes for this are glassy eyes, coal-colored bodies, and striped thorax, which are the recessive, and then they each have their own wild type, which we do not put in the graph. So I determined the allele arrangement. I looked at the two most popular, which was glassy eyes and striped thorax and coal body, and figured out that it was glassy eyes, wild type, striped thorax, and wild type coal body, wild type for the other. And then I looked at the two least numerous, which was glassy eyes and coal body and striped thorax. And I determined that the allele that flipped was the glassy eyes. So that's what I got for the heterozygous parent and the recessive parent. And then from coal body to the glassy eyes gene, I got 11 plus 8 plus 29 plus 27 over 1,000 times 100 is 7.5 map units. And then this was the tricky part for me. From the glassy eyes to the striped body, I didn't see any, what do we call it? Phenotype for as if striped thorax. So I was looking for like just glassy eyes, which isn't on here. And I was also looking for a coal body striped thorax, which isn't on here. So I got 0 for both of those. So I put that in, and I got 1.9 map units. And then I put the graph, and my interference came out really weird because I did 0.075 times 0.019 equals 0.00143 times 1,000. We should have expected 1.43. But 1 minus 19 over 1.43 is negative 12.3 times 1,000 equals like 123%, negative 123%. So not sure what happened with that problem. For number 4, just like the other ones, this one is anther ear, Bradick, and a fine are all found on chromosome 1 of maize corn. Then those are all the recessive genes. And then it just wants us to figure out the arrangement of these 3 genes. So I looked at the 2 most numerous and found brachytic and fine. So that would go with the wild type for the anther. And then anther, which goes with the wild type for brachytic and fine. Then I looked at the 2 least numerous, which was brachytic and anther and fine. So I could tell that the fine gene switched, so that would be in the middle. So it would look like this part. It'd be like anther, fine, brachytic, and then in that order. But we'd have the wild type for anther with 2 recessive, and anther with the 2 wild type for fine and brachytic. And I did the same process to get from the anther to the fine gene. It's 2 plus 3 plus 87 plus 98 over 1,000 times 100, which is 19. And then from the fine to the brachytic, I got 2 plus 3 plus 26 plus 20 over 1,000 times 100, which is 5.1 map units. I drew out the chromosome. And I got 0.19 times 0.0151, which is 0.00969 times 1,000, which is 9.69. Then we put that in, and 1 minus, we saw 5 over 9.69 is 0.484 times 100 is 48.4% interference. And then number 5, the one problem that was actually different. Alright, so it says suppose the cell has the genotype A, or dominant A, recessive A, dominant B, recessive B. Assuming that the A alleles are separate from the B alleles on different chromosomes, separate homologous chromosomes, what are the genotypes of the gametes produced by the cell, and what is the frequency? So I just said this, like sorted independently, so I would get 2 dominants, 2 recessives, and then the 2 different heterozygotes, and they're each equal in chance, so they all have a 25% chance. The next part said, suppose a cell, same case, except A, the dominant A is linked to dominant B, and recessive A is linked to recessive B. Assume that crossing over does not occur. So if crossing over doesn't occur, then that means that A is going to, the dominant A sticks with dominant B, and the recessive A sticks with the recessive B. And your gametes are 50-50. For part C, it says, suppose a cell has a genotype dominant A, recessive B, dominant B, recessive A. Again, A is linked to B, and AB is linked to B. What are the genotypes produced if the cell crossing over occurs? If crossing over does occur between the AA alleles and the BB alleles? So for this one, it says crossing over does occur, not that it may occur, which means it's going to have to occur. So if it's going to occur, then that means the dominant A is now with the recessive B, and the dominant B is now with the recessive A, because we know it's saying that the crossing over is occurring. So I got it flipped. So recessive A with dominant B, and then dominant A with recessive B. So it's 50-50, because it says that the crossing's over occurring, not that it has a chance to. And then for part D, it says, Suppose that the distance between the A alleles and the B alleles is 7.8 map units. What is the frequency of each gamete produced in the cell? So 7.8 map units also means that there's a 7.8 chance that crossing over does occur. So I pretended like the two heterozygotes happen 7.8% of the time, but I have to divide it by 2, so I did 3.9% for each. And then I did 100 minus 7.8 and divided by 2 for the two homozygous, or 2 dominant and 2 recessive, and I got 46.1 and 46.1. And that's how I did the genetics problems for this week.